Hello and welcome to this Introduction to Microbiology video. I'm Dr. Aled Roberts, and in this video we're going to look at the basics of viral replication, the different stages and the likely outcomes for host cells following infection. And so I guess one of the first questions we must ask ourselves is, how do viruses know what host cells to infect so that they can replicate? Because we have to remember that they cannot do anything by themselves. They require a host cell to complete various biological functions. And so to answer the question, we can rely on some basic biological principles. The first of which is the susceptibility of the host cell. For a virus to infect a target host cell, it must have a receptor that matches a protein on the surface of the virus. Now, in terms of susceptibility, this means some viruses only infect certain hosts. So for example, some viruses only infect humans, whilst others infect animals. And this is because only certain hosts have the correct receptor for that virus. And it's not just different hosts that have different viral receptors. Different tissues and even different cell types within a single host can display different receptors, meaning viruses tend to be quite specific in the cells that they infect. Now, the second aspect of viral infection revolves around permissiveness. This is based on the idea that whilst a virus may be able to enter and infect a host cell that displays the correct receptor, it also needs to contain the machinery that allows the replication of progeny virions, because without this, the virus would simply infect the cell and nothing would happen. Now, both factors are crucial for determining the outcome of a viral infection and supporting viral replication. And so when we look at the replication process for viruses in more detail, we see that it's a complex 7-stage process that is referred to as the burst concept. More on this later. But it differs from processes like binary fission, which is specific to bacteria, or even mitosis and meiosis, which are associated with our eukaryotic cells. Now, this 7-stage process follows a set order, starting with the attachment of the virus to a susceptible host cell. The virus then penetrates the host, whereby it undergoes an uncoating process, releasing the genetic material of the virus into the host cell cytoplasm. If the host is permissive to the virus, the genome will replicate and viral proteins will be produced through multiple different biosynthesis pathways. Everything is then assembled within the host, where the virus undergoes a maturation process before hundreds of progeny virions are released from the host cell, ready to infect another. So let's have a look at this in a little more detail, starting with attachment. Here we can see our virion, which are complete infected virus particles approaching the surface of a susceptible host cell, meaning they have the required cell surface receptors to allow for attachment. And so they bounce across the surface of the cell until a process of complementary binding occurs, where a specific protein on the virion binds to a specific receptor on the host cell. Once this attachment starts, the complementary binding usually instigates some form of conformational change, resulting in two different types of penetration. The first method is injection. This is where the genetic material of the virus is directly injected through the plasma membrane into the host cell cytoplasm. The second method is known as endocytosis, and this is a little more complex. Essentially, we have the same attachment process between viral protein and host receptor. However, in this instance, it signals to the cell that whatever is attached to the receptor needs to be ingested via an endosome. And so to achieve this, the attachment of the virus will signal to start the endocytosis process. This is where the surface of the host cell starts to invaginate, folding inwards around the virus until it's fully surrounded, eventually pinching off to form a vesicle known as an endosome. And what this does is it places the virus within a membrane-bound compartment within the cytoplasm of the host cell. Now in this state, the virus is actually hidden from the host cell, All the host cell can detect is the endosome, which is essentially made up of the cell itself, and this allows the virus to travel undetected within the host cell. Now the next phase is the uncoating process. This is where viruses not capable of direct injection, so those that have undergone this endocytosis process, release their genetic material into the host cell. Now this is usually the result of a mistake of the host cell. You see, when foreign objects are taken into the cell via endocytosis, a lysozyme will attach to the endosome and release lots of different digestive enzymes into that endosome to break down what has been engulfed as a defensive strategy. However, for many viruses, this allows the viral genome to make its way into the host, flipping the defensive strategy against the host cell. And so at this point, regardless of the penetration strategy, we end up with the viral genome within the host cell. And through various mechanisms, the host cell will synthesize viral proteins using viral genome as an instruction set. Now obviously, the route to producing viral proteins is dependent on the state of the viral genome and whether it's made from DNA or RNA, whether it's double-stranded or single-stranded, and whether the strands are positive or negative sense. Now if we do look at this in a little more detail, viral proteins are created using positive sense viral messenger RNA using host ribosomes and the process of translation. However, in order to get to this point, the viral genome needs to, at least in some cases, depending on the type of virus, undergo transcription using viral or host polymerases. And so in doing this, it completes part of the biosynthesis process and allows the production of things like the viral nucleocapsid and various other viral proteins. However, each new virus will need a copy of the viral genome, and this is completed via the process of replication, again using either viral or host-derived polymerases. And so at this point, we have everything we need to make new progeny virions. And so we enter the assembly phase. Now this can take place in either the nucleus or the cytoplasm, with DNA viruses typically being assembled in the nucleus and RNA viruses being assembled in the cytoplasm. Now once all the virus proteins are fully assembled, we have what we call a virion, which is our complete infectious virus particle. Now some viruses, once assembled, undergo a sort of maturation process within the host cell so that they can be released into the external environment and infect other host cells. And so in order to exit the cell, they enter the release stage, which is the final stage of viral replication and has two general mechanisms. First up, we have the destructive release of the virion, where the host cell is lysed as the virus exits, spilling the contents of the cytoplasm into the external environment, resulting in host cell death. The alternative release pathway is the non-destructive release of the virion, where the host cell remains intact, and the virus achieves this usually by taking some of the host cell membrane with it as it exits, essentially becoming an envelope virus in an almost reverse reaction to what we saw during cell entry. Now, the dynamics of the viral replication process differs to other replication processes that we see in microbiology. So as you will have learned elsewhere, binary fission in bacteria starts with a lag phase, where the bacterial cells double in number. However, because the starting point is very low, it takes a while before the number of cells increases logarithmically. That is, until the environment is unable to support the continued growth rate. And at this point, the bacterial cells will enter a stationary growth phase where the number of cells growing and reproducing equals the number of cells dying. And so if we compare this to our viruses and the burst concept, we would actually see a decline in the number of virions present in a sample early on. And this should make sense based on what we've just learned. Once a virus has entered a cell, the number detected in the environment effectively decreases. Following on from this, there is then an eclipse phase, which is where the progeny virions are being produced within the host cell. Finally, we then have a burst phase, where there is a rapid release in the number of detectable virions as the host cell releases hundreds of virions into the external environment in order to infect susceptible and permissive host cells. And with that, we come to the end of this video. Hopefully you found the content useful, informative, and most importantly, easy to understand. Thanks for watching, and I hope you have a great day.